Hello everyone, welcome to another practice session. The topic for this session would be mol bio, and this is the session 2. You can always go to session 1 and the link is given in the I button. Now, these kind of sessions would help you for IIT JAM, GAT-B, CSIR NET, GATE, these kind of examinations. I'll also tell you what resources you can possibly use to get more such questions. There would be 5 questions and we would take 10 minutes to solve these 5 questions. You can solve with me. Now, these are the important topics which are crucial for any MSc level entrance examination. And today we are going to discuss about molecular biology. Apart from molecular biology, if we only talk about GAT-B, there are 3 more topics which are newly introduced, like bioinformatics, bioengineering, and cell culture methodologies. If you need help for these chapters or these topics, then you can join unacademy's Synapse batch, which is starting from 28th April 2021, and this would be dedicated for GAT-B 2021 preparation. And by the way, there is 4 days to go for the price hike, and if you don't subscribe quickly, then this course would have a significantly higher price. So hurry up. All you need to do is study from home, and you can study safe, and it's cost effective. Believe me on that part. You just download the app from your app store, use my code AP10, and then you can start learning from the platform, from all the educators, and they would guide you throughout this process, and it would make your preparation easier. Okay, enough of this promotion. Now let's talk about the questions. Okay, this is one type of question which is very common, and the variant of this question is asked in several exams. So here it says, which of the mRNA will be translated into a polypeptide chain containing 8 amino acids? So we know in the open reading frame there would be codons, and we have to understand which of these sequences can possibly give rise to 8 amino acid containing peptide. So you have to first understand how translation occurs. So it would start from a start codon, and if you see all the sequences, you see AUG in the beginning of all these sequences. So here is the start site, and after that the translation would stop where the stop codon is, right? So look for the stop codon sequences in all of these options and where you see it. Okay, if you have done that successfully, look at this part, right? So here you see the stop codon sequences. So right now, if you count here, 1, 2 amino acids, 3, 4, 5, 6, 7, and including AUG, it would be 8, right? So B would be the correct option. Okay, next question says, arrange the following events in the order of synthesis of proteins. Peptide bond forms, a tRNA matches its anticodon to the codon of A site. The movement of second tRNA, the second tRNA complex from A site to P site. Then the option says the large subunit approaches the small subunit and the initiator tRNA fits into the P site. A small subunit binds to the mRNA. The activated amino acid tRNA complex attaches the initiation codon of the mRNA. So we have to understand that what are the sequence of events, right? So first thing first, the small subunit would attach to the mRNA, and you know about the Shine-Dalgarno sequence or the CUCET sequence, right? If you don't know, you can get the video and the related concept in the I button, so take a look at that. And then the first codon, the initiator codon, there would be a tRNA which binds to that codon, and ultimately then the large subunit would associate. So these are the key events, right? And all these events are nicely described in this video which you can get in the I button. So this seems to be the correct option. So option D is correct here, right? Question number 3. Again, this question is very, very famous, and these type of questions are asked all the time, right? So here you see a bunch of enzymes and their function, and you just need to match the following, right? And for these particular question, I'm not going to answer, and I'm going to ask you to give your answer in the comment section, and I'll say whether you are right or wrong. And you can also understand, right? Okay. Question number 4 says, in an experiment, you wanted to see which mutation will have worse effect on transcription. In the first set, you have deleted 94 amino acid from the C terminus, and in the second set, you have replaced 265th of the C terminal arginine to cysteine. In your opinion, which would have the worst effect? Options are 94 amino acid deletion mutation, 265th amino acid mutant, or both would have similar effect, no visible effect on transcription. So what would be the important funda here? So we know that in the eukaryotic transcription, the C terminus of RNA polymerase, C terminus of the RNA polymerase would have a heptad repeat, right? So these heptad repeat has specific residues which gets phosphorylated, right? And these residues need to be phosphorylated in order to start the transcription. Now, if you delete 94 amino acid from the C terminal domain, right, then the chances are your heptad repeats are also gone. So that is why this particular mutant would have the worst effect. If you don't know what is the eukaryotic transcription process, you can always go and watch the lecture in the I button. So the link is given in the I button. Last question of the day. Which statement is false regarding histone? In eukaryotes, histone are poorly conserved. There are at least 5 different classes of histone. Histone in tetrahymena macronucleus have very high level of acetylation. Histones are richer in lysine and arginine residues than most other proteins. So we have to identify the false statement. So let me tell you, this is the false statement, because in eukaryotes, histones are widely conserved, and there is a histone fold which is conserved throughout the eukaryotes. So this statement cannot be correct, and this is the answer. And do let me know your answer to the question number 3 in the comment section, such that I know. If you like this video, okay, so there is a question number 6 before saying that golden word. Question number 6 is for you again. A researcher found a method that she can use to manipulate and quantify phosphorylation and acetylation in embryonic cell culture. In one set of experiment, she succeeded in decreasing the acetylation of histone tails. Okay, which of the following result she would mostly expect? Okay, increased chromatin condensation, decreased chromatin condensation, decreased binding to the transcription factors, inactivation of the selective genes. So this is another question, question number 3 and question number 6, which you can possibly answer in the comment section. So do let me know your answers in the comment section. And if you like this video, give it a quick thumbs up. Don't forget to like, share and subscribe. Thank you.